this particular chapter. Thank you very much. And I'll kind of take you where we left off. There's a great deal of complexity that's coming your way. Okay. I mean, Monday was mostly terminology. Now we're going to really get starting into the complicated nature of it. I'm going to probably show you an animation shortly and then get back and discuss it. Maybe the way to go. And then we actually view that animation again. This is the beginning to understand this whole concept of how this sarcomere shortens, which is effectively how muscles move. And as these sides come together, okay, individually, and the thousands of them that are throughout any given muscle, okay, and effectively all it's doing is you're going to learn where we're going to watch these guys in blue who are called actin slide over these guys in sort of this reddish color, which is myosin. And effectively, the sum and substance of all this, we add up all that shortening that goes on, pulling the boundaries, the Z discs, slightly closer to one another, and effectively that's what creates motion. The whole idea is called the sliding filament theory, and again, this is basically how muscles move. So you're going to see this. There's a ton of information, and that's why, again, I make this a standalone chapter. And I mean, I had a lot, and in this early days of going over it, there'll be a lot of blank stares. The reality is, for most of you, this is not something no one is ever going to ask you about, but it becomes one of the tests and why it's in a curriculum. To just understand, why do we learn stuff that we're probably never going to be asked about? No one ever asked me about this, and I'm a doctor. I treated patients for 25 years. It's the ability, and it's part of the curriculum, to be able to assess, in this case, quite complex information and be able to have what we would describe as a passing knowledge or understanding and be able to answer questions to a passing knowledge or to a passing degree, whatever that is arbitrarily, that figure. So, I mean, it's sort of like a test of being able to assimilate difficult information, have a given level of understanding, and be able to respond to it. That's really what a lot of this is about. I mean, and really it serves you very well for many of you, particularly in nursing, going into pharmacology and pathophysiology, which are very, very difficult subjects, really advanced subjects. That's kind of where this all comes from. So, I mean, you have to have some context, because I'm sitting where you're sitting, it's like, why, and not speaking the term that you might speak to each other, why in heaven's name do we have to know all this stuff? You can imagine the words that I would use. I have a son in the Navy. They speak fluently in profanity. And he's the boss. Okay. So what you're going to see is in these thick and thin filaments, this thick one is called myosin. The thin one is actin. You should be familiar with the name actin because all those neat little filaments that make up microvilli that allow endocytosis and exocytosis, allow amoeboid motion, allows those cells to scooch around, are made of this same material, actin, except in the cell, you remember the animation, we can build them up and take them apart. Okay, here, they're always there. They become a structural element and form a tiny filament, as you will see shortly. So these are actin and these are myosin, and for all intents and purposes, the actin, as you will see, get pulled toward the midline, and in doing so, bring these Z discs closer together. They do so because of these myosin heads. Just to oversimplify it, the myosin is a complicated protein that has a thick strand and a head. And that head, your muscles are all ready to fire. Here's the actin sitting almost on top of it. And when an area on the heads are finally exposed to one another, they're in such close proximity they touch, and effectively that triggers this. It's almost like a trigger ready to fire. We call it cocked anyway. And then all of a sudden it goes boom, and it pulls those actins all over the place toward the midline. That's effectively how this sliding filament thing works in a very oversimplified fashion. There are three excellent animations. We're going to look at the middle one today and then look at it again down the road. And that's why I spent, surely we're going to be spending time looking at this chapter, not just on Friday, not just the Monday and Friday of next week, but going on even into November. It's that degree of complexity that's involved. So the thick filaments are myosin. That's the name of the protein. The heavy chains, you don't have to know this. The heavy chains make up this sort of stem, and then the heads, these four light chains. And that we talk about heavy and light chains. It's not something you have to know. That's biochemistry. It's part of it. The heavy chains make up this sort of strand or tail, and the light chains make up the head. And what I was describing to you is this cross-linking or cross-bridging that occurs. And you'll see, interestingly enough, as you begin to look down the road at these animations, you're going to see the relevance of this being offset. Okay? That if you go back and look at that diagram, the heads are not all in alignment. Some are vertical, some are in varying degrees. They go all around and they're not all in the same place. Because you're going to see the difference between when you lift something light and you try to lift something that's heavy, you have to engage more power in your muscle. And by doing so, you're releasing and attaching these heads. You're continuously ramping them up to pull toward the midline. And what happens if you detach all of them simultaneously, you go back to your starting point. Not so. That offset means about half of them are released and the other half still become attached. And so it's kind of like doing a rope climb. If you let go of both hands, you're down. But as long as you're holding on, you're getting up. I was never any good at that in gym. My 5-year-old grandson, very good at that stuff already. So that's when we'll look at the idea of the staggering. That's about that. So the molecular composition, the thin filaments are made of actin, that same material. What we were seeing in the cell are changing between globular actin, which is soluble, dissolved in the cytoplasm, and when it assembles, it's called filamentous actin, or F-actin. That's where the little prefixes are. So those little subunits form this long fiber called filamentous actin. They don't dissolve. So in the muscle, we don't have to worry so much about assembling and disassembling. It's there. There's a permanency to it, unless the muscle was damaged, and then they wouldn't be there. And what happens is it looks for all the world like a double string of pearls. It's almost like you had a big string of pearls and you folded them a little bit, and that's kind of what it looks like. That's sort of the analogy I use. And what's interesting, as you will see, and you're just going to hear about it today, you're going to see it in action. My guess is maybe not even Friday, maybe sometime next week. You have what are called regulatory proteins. They're called tropomyosin and troponin. And what they are, they attach to the actin. You'll see the illustration in a second. So here's the individual head. And it has, on the end of it, it has binding sites. This is a little confusing for students. On myosin, we have what they're called actin binding sites, because they're not named for what they're on, they're named for what they're attaching. Think of the radial head of the radius and the radial notch on the ulna. The notch is named for what it's going to articulate. So we see this a lot. It's like the zygomatic process of the temporal bone and the temporal process of the zygomatic bone that unite to form the zygomatic arch, which you probably heard a little bit about in anatomy. I took that course too. So here it is. I told the other class, for the sake of full disclosure, I cheaped out. Normally I bring in peel apart Twizzlers and regular Twizzlers for this. Do you know what they're charging for Twizzlers? A bag of those things? $6.77 at Walmart yesterday. I'm sorry. I don't get reimbursed for the Twizzlers because they know I'll eat them. My wife said it's a good thing because I usually leave them for the students. You guys never eat them, and I end up having to eat them. You would have eaten them. How are you skinny? I go to the bakery and I smell things, I get fat. I have particularly the Oakmont Bakery, for you Oakmont Bakery fans. Oakmont Bakery fans? Oh. The guy who fixes my car, just I got it from him, is in Oakmont. Any excuse to go visit is fine. My daughter lives in Indianola, just across the river from there. We're going to visit Oakmont Bakery. Moving on. So this is what. So imagine a Twizzler, and it's tilted up, and that's the head that I was talking about. And these are the binding sites that bind to the actin. And actin, here are the individual actins, and they have, I know it's confusing, myosin binding sites, named for what they're going to attach to. They're sitting there on this strand, literally next to each other. The only thing that's keeping them from attaching and causing that muscle to fire is this. This is what a regulatory protein in tropomyosin is, a strand-like protein. And you can see what it does. It covers the heads. It's held in place, and really this is the analogy for it. I mean, these are exactly what the proteins are shaped like isn't clear, but they're effectively held in place like a thumbtack or a push pin, like you're pushing something over there by a regulatory protein called troponin. So imagine effectively if the active sites were like this, and we turn them around. But if I did this and wove them around, the active sites were on the edges. I mean, that's all well and good. But if I don't have something holding me in place, as soon as I would let go of them, use toothpicks. Effectively, the toothpicks, thumbtacks, the push pins, preventing that nonsense from happening are the troponin. That's the analogy that I hope helps you. That'll also be like similar to the analogy that would be on a test question, but don't worry about it now because you have a more important test coming up next week. Moving on. So then you can see that's the thing. And there's a very good animation that you will see that shows us the third of three animations that we look at to describe this activity. And so here you can see the individual subunits, but they're not separate the way we find them in skeletal muscle. They're like this. And so that's the molecular composition. We're not done yet. I showed you that elastic filament tight. And again, when it stretches and it recoils so it doesn't get stretched out, or what would happen with your injury, some of those fibers are going to be damaged. A question of a matter of degree. Okay? That type of thing. So recoil resists excessive stretching. We actually name one of these there's other proteins whose names you do not have to know. The only one that's of relevance for our discussion is this, called dystrophin. When someone has a disability, they lack an ability. If you have a dysfunction, you're unable to do a certain function. When we talk about, anytime we use the prefix dys, it means unable to, or doesn't do it properly, something's wrong. Trophy means growth. So dystrophin was named for the disease that it causes, called muscular dystrophy. They didn't have a name for the magical name for the protein. They named it off it because it alters the growth, or grows inappropriately or irregularly. So this is the most famous of the muscular dystrophies, although there are certainly more than one. This is called Duchenne's, the way I pronounce it, muscular dystrophy, the way I learned. Okay, most common, very, very serious, life-threatening, life-shortening, okay, that generally appear during childhood. Sort of like when I was describing Tay-Sachs to you, things look normal, and parents, again, normal, healthy baby, and all of a sudden, they're having difficulty standing up and difficulty walking around, or having any degree of strength. Because what happens, it's almost as though the muscles, rather than, they look like they're good and firm, but the reality, they're deteriorating inside and eventually replaced by like a fatty, deteriorative tissue, and really have very, very little strength. The bad news long term is it affects all muscles, particularly respiratory muscles. And so when you get into it, I know you learned about them in lab, there's internal and external intercostal muscles that allow you to breathe. Now, these children, when they get to probably before middle age, typically would succumb to respiratory diseases and eventually die from that. It's called sex-linked recessive. If you're unfamiliar with that, I know some of you have learned a little bit about genetics and Punnett squares and things like that. Sex-linked just means it's carried on the X chromosome. So sex-linked happens to be something, since it's on the X chromosome. In women, it's typically mass with two X's, but on the males, and basically it demonstrates itself. It's comparatively rare. That's not that terribly common. And you can see when it appears and what happens. Only that head, particularly chest muscles and cardiac muscle, that's the significant problems vis-a-vis. And we probably, since this is about an 8-year-old PowerPoint, I would say the numbers have increased. There's a lot. I explained to the other class. For years, the reason we didn't have treatments for a lot of comparatively rare diseases is there was not a financial incentive. Pharmaceutical companies are companies. In business, the bottom line is the bottom line, making money. So even though they're making things that help us live and survive, they're doing it. Mostly they would make things that people would use all the time. About not quite 40 years ago, in 1988, they came up with something called the Orphan Drug Act. And I talk about it again in micro. It has nothing to do with orphans. It has to do with a disease. It's a financial incentive for the development and bringing to market of medications for diseases that affect a limited population. It was brought about specifically for AIDS, because the community that had the most problems with AIDS and the most visible people who were affected were entertainment, movies, and things like that. Okay, and because there were a lot of, for lack of a better way to express it, closeted gays who were suffering with this particular disease, and there was nothing they could do to treat them once it finally reared its ugly head. And so it enhanced our understanding of HIV and AIDS and brought medicines to the forefront to be able to treat them when they were very, very limited. In micro, when I go over this, we revisit this again. So there's a lot of history, and that history has helped other disorders as well. This would likely be one. So this is the gene, and they named the gene for what it does, dystrophin, that links filaments. You don't have to know all this stuff, and stabilizes the membrane, and tears, blah, blah, blah. And inflammation, regenerative capacity, blah, blah, blah. Okay, so effectively what happens, the muscles don't work. Let me show you this illustration. And this is so it's very, very difficult. There's a number of diseases. For years, the only way they raised money for research was every Labor Day, they had a telethon, 24-hour telethon over Labor Day weekend that raised money increasingly over time. Started as a very small telethon and then became sort of a Labor Day staple. And they'd have, a lot of entertainment people would come, and they try to raise money. And that's the way it worked. And so that's the best I can tell you about that for now. The other thing you're going to see in the animation, as well as kind of giving you an idea of how all of this occurs, and it shows it very, very well on this animation that we'll see more than once. It's called the sarcoplasmic reticulum. That's code in muscles for smooth endoplasmic reticulum. Because they really don't have to make proteins, they've already made them. So really, effectively, the only thing they really have is smooth endoplasmic reticulum. And they have little well-like cul-de-sac structures which hold calcium. That's their job. And calcium is critical because the ability, these cells store calcium, and they store the calcium in such a way that the release is triggered by an electrical current that passes through it. So you're going to see, without going crazy about it, you're going to see this sarcoplasmic reticulum enveloping an individual muscle fiber and all of its fibrils that are there, mostly running the long ways. And really, and this is, again, not a big deal for you, at the A and I band junction, you're going to see what's sort of a neat kind of presentation where they have these cisterns. A cistern is a well. So these wells are these cul-de-sacs with calcium. And when they talk about regulating intracellular calcium, basically it stores it, it holds it, and releases it. In skeletal muscles, the calcium is already in the muscle. In cardiac and smooth muscle, we talk about extracellular calcium. This is your first opportunity to see the extraordinary importance of ionic calcium. And at skeletal muscle, it's already in there. The calcium in the bloodstream isn't a big deal. When we talk about cardiac and smooth muscle, it is a very big deal. So so you really have to know your calciums. And we're not done with calcium, because in order for the nerve to send that signal that'll make that muscle fire, we got to have calcium, extracellular calcium as well. So you're going to get a whole lot of terminology that's going to drive you a little bit mad, I anticipate. Here's some more. Big T stands for transverse, and you'll see this in the animation that I will show you. And these are part of the cell's membrane, also called the sarcolemma. And what they are going to be married to a lot of that A and I band with the terminal cisterns. You're going to see it. And this is the way the electrical signal, which is on the outside of the muscle fiber, gets inside. And these form, the way I would describe it, are perpendicular invaginations. I know that's heavy-duty language, but you're going to see it better when I show it to you. Okay? And what it does is, really the bottom line is transverse, because they're going at right angles to the long axis of the muscle fiber. And this is the way the signal is transmitted. And what happens, they form something called a triad. And you'll see this again in the animation. We're going to have that portion of the membrane coming in at a right angle. On either side of it, you're going to see one of these wells, and that's where the calcium is stored. And as the electrical signal passes, it's like flipping a switch. It's got a fancy name. It's called a voltage-gated sensor or a voltage-gated channel, and you're going to get sick and tired of hearing me talk about them, because we're going to talk about them a lot over the rest of this class. And this is your introduction to it. And they form something called a triad, because you have a T-tubule, transverse tubule on either side, surrounded by these well-like structures called terminal cisternae. Cisternae is the plural. So that's the triad relationship. The tubule proteins act as voltage sensors. And when they're triggered, they change shape, and it allows calcium to flood. So the electrical impulse passes by, the proteins change shape, and the sarcoplasmic reticulum proteins are also change shape, and the calcium is released into the cell. The calcium is going to attach to that troponin, the thumbtacks or push pins, change its shape. And do I have to do that again when it slides off? No. I know. Why didn't you get the Twizzlers? Because I'd eat them. For that, we use peel apart Twizzlers. There's a Twizzler distinction. Let me see if I can get you to this animated before we go through all that nonsense. I'm probably sick and tired of seeing it. Let's do the animation so you can see what I'm talking about. There's a lot. I'm not even touching this stuff yet. As you can see, there's a lot going on here. This is the first animation. Look at all that good stuff. This is what we're talking about. It's called excitation, contraction, coupling. We're going to go over all of this again ad nauseum at length. Voila.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So, I mean, this is what we, and so I think and again, these are very high quality. And we're going to talk about the signal and how it gets there and how the signal gets through. That's coming up. And that's really the next major, before we return to this. But this is a really good illustration, as you can see, of that's what these transverse tubules are. Regularly spaced means they're at every one of these A and I band junctions. And you can see this is how they penetrate. This is electrical current going through. And because of the unique arrangement, these T-tubules, and they use a great term, abut, they basically are surrounded by these wells. And here you can see a well on either side. Those are, again, cistern is another name for a well. So they're called terminal cisternae. And where they contact each other on either side, there's three things. So it's called a triad relationship. Triads are three things. And then you can see as that light goes through, basically that's the electrical signal they're representing. And what it does, this is a voltage-gated sensor. And so we call them voltage-gated channels. Because the channel, once voltage goes by, will effectively create an opening in the cistern that allows the calcium, you can see these tiny little red dots, ionic calcium flooding the area. They're going to flood and attach to the troponins, release them, and effectively, as long as they're there, we're going to be continually causing that muscle to contract. Why do we have to contract it more? If we're lifting something heavy, whether lifting something light, if we have to generate more power. And effectively, we're going to get to all of those things as this sort of discussion takes shape and this chapter gets covered in really a fairly significant degree of detail over time. And you can see it basically compressing on itself, and that's the next part I'm going to show you. I'm going to go over this one again, and that's what I want you to do. Oops, sorry. This is what I would like you to be able to do, is to go back and look at these things. That's why I saw it the other class, I'll say it to the next class, I'll say it to my Wednesday afternoon class this afternoon. I record these so you can listen to them, not just listen to the review. I was disappointed that people weren't doing it as much. And inevitably, the people who are here are not the people I've missed disappointed. But I think it's a useful way for complicated material to do it. I don't know of any other professors who do that stuff. But when we started doing this during COVID, I just figured it was a good idea. I convert them to YouTubes, and Lord knows, if you're anything like my kids and grandkids, you're YouTube freaks. Just everything is right. My 82-year-old brother-in-law watches YouTubes all day so he can work on his golf swing. I was somewhat astounded by that. Just to let you know, I'm just not the old oddball. My wife watches them all day too. I'm living in the past, Lou. So here we are. Let's go. Here's the T-tubules. Here's the triad. That's right. So this is what, and now the schematic makes sense. Again, the fact that it's on the A and I band junction, who cares? Really not that important. But effectively, there's a whole lot of these voltage gated sensors or channels, and it allows the calcium to flood in. And effectively, it's the summation. It's all those little sarcomeres being pulled. And again, the way we do this is not a fold. Okay, this is these filaments, the actin sliding over the myosin. As the myosin, what happens to calcium effectively triggers those myosins. It exposes those heads, those active sites I talked about, to one another. And they're so close, immediately they bond and it fires. And they continue to fire and reload. You're going to see the amount of energy that's involved. That's why our muscles, the vast majority of our ATP, okay, other than the part that's using to continually reset nerve, basically it's all dedicated to resetting nerve and muscle cells, because we use them all the time. You don't have to be using them, like now you're not using your muscles a heck of a lot, but you're hopefully thinking. Maybe you're not thinking about what I'm telling you, but that's okay too. Nobody skipped more classes than I did as a freshman. Nobody paid less attention. It was fun. Believe it or not, we could smoke in the classroom. I mean, it was a different world 56 years ago, but shut up, nobody cares. Even the teacher, we had teaching assistants, grad assistants. He's teaching us English. I didn't know English, and he's got a cigarette. I figured I'll light up. At least it wasn't one of those unusual cigarettes, though that happened too. Remember I told you the Woodstock thing? You know, did you look it up yet? We've got to work on that. Listen to the music. It's really good. Oh, Lord, spare me. I'll have to show you pictures of the wedding reception for my daughter. A whole bunch of 40-plus-year-olds acting like they're your age. Trust me. I think alcohol had a significant influence on that, but I digress. Okay, so the cross bridges are what generate the force, and the shortening occurs basically, and the contraction ends when the cross bridges get inactive. We're going to see all of that. So the thick and thin filaments overlap only slightly. This is the idea of the sliding filament theory. It states that contraction, the thin filaments slide. You're going to see it here. Graphically, it's very easy. Neither thick nor thin filaments change length. That's the misconception. They're not bending. They're not stretching. They're just sliding over one another. So the nervous system sends a signal basically saying, okay, I need to move this muscle. The myosin heads are allowed to bind to the actin. That's where they release that calcium. And cross bridge. And yes, I know this is a lot to digest in one class. We're going to go over it a zillion times. Please. So the cross bridge attachments form and break. And again, that's, do I have to lift something fairly easily, or do I have to put a lot of force into it? Pulling them closer and closer together. So what happens, the shortening is really the Z discs in the individual sarcomere getting pulled toward the midline. You're going to see the I bands appear shorter. They are narrower, if you want to think of it that way. The Z discs comes together. The fact that the H zone disappears and the A band moving is not so much. This is a micrograph, and this is the schematic, the illustration that we saw before. Notice between the two blue, this is how far that can contract. If it's going to maximally contract, the overlap is going to take this actin in blue all the way to the midline on both sides. It will do that by effectively what I call ratcheting or sequential firing of this. It's like that, okay? And we'll bring that to its maximum degree of contraction. What you want to look at is the boundaries. Here's the I band, light. Here's the A band. It's really not going to change. It's going to change color a little bit, but not going to really change size. So if I do it here and I advance to the next slide, look at the cursor. If I can keep it here. See that? Now look at the I band. Much, much narrower. Agreed, the A band has a little bit of a different color, but look where these things are. We've effectively, why it's fully contracted, we pulled these. We don't have to fully contract every time. all the way to that mid zone. So the A band effectively is the same, okay, if you want to think of its girth, but the I band, oops, went the wrong way. The I band is wider. So I was over here, and now let's go forward. You see what happens. Here's the A band. Here I'm backspacing it. Now, but look at the I band. So really the shortening is occurring with roughly a maximum contraction, at least in this particular sarcomere, about halving that I band, and it's having it on both sides, as you can see. Both sides. So that, just think of the additive effect of not one or two, but thousands of these. And that's how many there are along an individual muscle fiber. And we take all those individual muscle fibers, and you'll see that we stimulate them in such a way as to not fatigue them. And that's how you generate it. I mean, and all of the understanding of this has helped exercise science, has helped physical therapy, has helped rehabilitation, understanding how muscles work, how to work on them, what helps them, etc. So the rest of what we're going to cover going forward is basically how this all event occurs. That's really what kind of happens when we're thinking about mechanically of that sliding filament. But we have to go into 3 different phases. The hardest one, I think, and the most complex one, is understanding how the signal gets from me. I'm going to move. I'm thirsty. I'm going to grab this. And all the steps are involved. And you're going to see it's very, very complicated. Okay? We call that the events at the juncture point between the nervous system and the muscular system, called the neuromuscular junction. Now, once we made the decision, caused that signal, then you just saw how we changed that signal from effectively electrical signal to mechanical action, excitation, contraction, coupling. And then if you want to really see how the contraction occurs and understanding how we're able to basically how it gets shorter and shorter, we call that the cross bridge cycle. Those are the 3 giant events, all of which have multiple steps. And this is the kind of question you get. We do things, and maybe you saw them in A&P lab, I don't know. We do sequence questions. It's like studying the sequence of events that occurs. And they're very, very challenging because it's like you have to put them in the right order. Does that make sense? So, I mean, for instance, in lab, the typical question will say, okay, trace a drop of blood from where it leaves the heart by way of the aorta till it comes back by way of the either superior or inferior vena cava. So you follow the sequence. It goes from the aorta to a particular artery, blah, blah, blah. That's a sequence question. How does urine form? That's one of the ones I utilize in lecture or in lab, is how basically it comes from, it forms in the kidney and by way of certain structures, and then goes from the ureter to the bladder to the urethra and out. So you have to understand the sequence of structures it passes through. This is the sequence of events. In a sort of a long-winded way to give you an idea of what's going on. So my decision to move that, well, I feel like having a drink, or I'm tired of talking, my throat's killing me. Okay, that's a decision activated by my brain. Now, how does it get to where it's supposed to? You'll come to know down the road it's a 2-nerve circuit. My brain telling my arm, hand assembly to move. So the first nerve goes to the spinal cord, to the appropriate area, and the next nerve tells the appropriate muscle or muscles to fire. 2-nerve circuit. So that decision is transmitted, in this case, down the spinal cord, and to nerves that are in the spinal cord that then go to the appropriate muscle fibers. Nerve and muscle cells, by definition, are excitable. That means that impulse triggered by my brain, that electrical signal, will rapidly conduct itself down the nerve, trigger the next nerve in sequence, and the way it triggers from nerve to nerve or from nerve to muscle, okay, are by chemicals. Those chemicals are called neurotransmitters. You're only going to have to know this one for now. But, oh boy, when we get to the unit, when we start talking about the central nervous system and the autonomic nervous system, there's a whole lot more neurotransmitters to know what they do. And thankfully, this is the best one to know first because it's the most abundant. It's the one we use the most in our body. Nearly all of the autonomic nervous system, which we cover as the last unit in AP1, it's in the brain. It's in every skeletal muscle the way it's told to fire is by a nerve releasing this chemical, acetylcholine, which you'll come to know is made of effectively vinegar, acetic acid, which is a lot of it in our body, not as vinegar, but it's something called acetyl, and a chemical choline we use all over our body. And we marry the two together. It's a very unique neurotransmitter. It's in a category all of its own. It's the one they first discovered. It's the one we understand the most. It's the one that controls your heart rate, for instance, if you didn't know. So just to give you an idea of the relevance of what we're talking about, this is why it's so important to understand these. Most of the medications that we prescribe to people, certainly when you're talking about somebody with depression, eating disorders, psychological or psychiatric disorders, what medicines all affect neurotransmitters, period. This is how the medicines we utilize, for the most part, control a lot of neurotransmitters. We're going to talk about heart medicines a little bit. We'll talk about blood pressure medicines. They're all involved sort of in a roundabout way with the understanding of this whole concept. So that may give it some context why we study it. I know it's hard. I understand. Right? This is my task and your task. So the nerve and muscle cells are excitable. They change that resting membrane potential, the sodiums and the potassiums, the minus 70 millivolts. They have the ability for that to rapidly change across the membrane and cause it to fire. Most cells don't have that. They all have a resting membrane potential, but these ones, it can change rapidly. That's what the definition of an excitable cell is. And it's all about, in this particular instance for skeletal muscle, acetylcholine. There are different channels. The acetylcholine is a chemical. So what does it open? This is where the terminology is a pain in the neck. Chemically gated. The other name we give it is ligand gated channels. You can use those terms interchangeably. Textbooks will, the PowerPoints will, anything you study about it will. So you have chemically gated or ligand gated channels, and the chemical messengers, neurotransmitters in our case here, like acetylcholine, will effectively open those doors. And voltage gated, I just explained to you. Voltage gated, they open or close in response to changes in membrane potential. How that rapid firing change in the membrane is what's like flicking a switch that I demonstrate. I'm happy I can snap my fingers today. When you get old and you get arthritis, it's not so easy. It's okay. I feel like something from the original Addams Family show from 1966. You watch that in Prime. Great John Aston, who is still alive, by the way, in his 90s. Voltage-gated ion channels. I always liked Uncle Fester myself. Should have been the back. Jackie Cooper. Voltage-gated ion channels. Open or close in response to voltage changes. Here's what they look like. Okay? Chemical bonds, door opens. Electrical signal comes across, door opens. We'll get to that bad boy down the road. That's called an inactivation gate. That's kind of like the key. Okay? And so without going into it, we're going to stop here, but let me show you all that's involved. We have to go over the anatomy, how the circuit works, all the different things that are part of it, and then all the events that allow this to occur. How's that? That's a whole effing S-load of stuff. Not to be critical. Goodbye.